Hi, hello. So today will be the final lecture on green alga. We have seen life cycles of Chlamydomonas, Chiara, Eidogonium, Volvox and others in our previous lectures, and this is the final on that series. Today we'll be finishing Spirogyra with each and every details required for the examination purposes. So what is Spirogyra? Spirogyra is a free-floating freshwater green algae which is also known as water silk or pond silk. Why it is called silk? Because it is having a silk-like appearance. Okay, so it is a green alga. So it is photosynthetic because of the presence of chloroplast in its body. Then it will be contributing to the carbon fixation, supply of oxygen to the aquatic medium. These things you can write in general. Coming to the structure of the Spirogyra, we can see that it is an unbranched filamentous structure. You can see the cells arranged end to end in a uniseriate fashion. In a single line, you can see that cells arranged end to end. Have you ever seen such a structure in any of the life cycles that we have seen? Yes, you are right. In Eidogonium, we have seen such a structure when unbranched uniseriate end-to-end connection of cells. So similarly, and in the case of Spirogyra, you can see different things. If you are having a diagram, this diagram you can easily make with pencil. Okay, I have attached only very simple figures which could be easily reproduced in the exam within no time. So from the diagram itself you can write the explanation. You can see that there are strands of chloroplast and around 1 to 16 such strands could be seen in a single cell, and within the chloroplast strands you can see pyraminoid bodies arranged in a row in each strand. These are for storing starch, proteins, etc. and other photosynthetic products. Then the area that is between these particular chloroplast strands and all is the protoplasm or cytoplasm of this particular cell. Then you can see that the in between the protoplast and the chloroplast you can see a vacuole present. These empty areas, okay, will be vacuolated areas, and outside the vacuole only you can see the cytoplasm or the protoplasm of the cell, and part of that cytoplasm you can see a nucleus present. So this is all about the particular cell structure of Spirogyra. And one more thing, you can see a mucilaginous covering outside the cell wall, and it is made up of pectose. This entire cell wall is made up of two things, an outer layer made of pectose and the inner layer made of cellulose. And the thing is that this outer pectose layer will be dissolved in contact with water, and it will be acting as a mucilaginous matrix around the cells of Spirogyra. So this is about the general thallus structure. You can write a very good paragraph about the introductory remarks or the salient features of Spirogyra with the diagram itself. Now moving on, this is another kind of figure which you can easily differentiate each of the parts. The mucilaginous sheet, cytoplasm covering the surface. You can see that just below the cell wall you can see the cytoplasm, and within that you can see a vacuolated area. These empty spaces are actually vacuole, part of a vacuole. Then you can see the cytoplasm and the cytoplasmic strands. Within that a nucleus is embedded. Then chloroplast and pyraminoid bodies arranged in a in a row. Okay, so this is about the structure that we have already discussed. Another kind of structure, you can see that this chloroplast is actually ribbon shaped. Okay, these strands are actually ribbon shaped, and it is arranged in a spiral manner. That's why this particular genus has got the name Spirogyra. Now coming to the systematic position. Whenever you are writing an answer on algae, you have to write the systematic position, as I have already mentioned this thing. So it's in the domain of Eukaryota, kingdom Plantae, then phylum Chlorophyta, class Zygnematophyceae. So this is something different, Zygnematophyceae, order Zygnamatelis, family Zygnemataceae, genus Spirogyra. And within the genus you can see different species. Around 300 to 400 species are there, out of which around very few are found in India. Fine. So this is about Spirogyra systematic position. Yeah, this is yet another filamentous structure. You can easily make by seeing this kind of simple pictures. Okay, yeah, the structure of Spirogyra I have already explained everything, I guess. Then one more thing, it is actually around 10 to 100 micrometer in width. Okay, that much small in width, its horizontal width, and it may grow several centimeters in length, not in meters length, several centimeters in length. Okay, thallus is multicellular with each cylindrical cell joined end to end. Okay, it's kind of rectangular cylindrical cells joined end to end in a uniseriate unbranched fashion. So that's it. Now coming to the reproductive details. So reproductive details of Spirogyra. This involves three kinds of reproduction. One is vegetative reproduction, then asexual reproduction and sexual reproduction. Out of this, asexual reproduction is actually less common in Spirogyra. It is found in very few species, and under very extreme unfavorable conditions you can see these kind of structures. Then sexual reproduction is the most common thing. Now coming to the details, vegetative reproduction is commonly done by fragmentation. How the fragmentation will happen? Maybe due to mechanical injury or dissolution of this middle lamella. You can see that this cell and this cell are joined end to end because of a connection known as like middle lamella, which will be the connecting like element between the cells. So this middle lamella from this end and this end may be dissolved, and the fragments may break like this. And from this broken up fragment, that will be doing multiple divisions. Each fragment, if this fragment is broken, that will be undergoing multiple division, and new filamentous thallus will be formed. Okay. So the filament consists of many identical cells. This is one cell, this is one cell, this is one cell, each capable of living independently. When this is broken, it will be dividing, multiplying, and it will be regenerated into new thallus structure. So this is the easy to draw diagram for the examination. Here there is a septum, and from here a fragmentation will happen, and this is the independent daughter cells from which new Spirogyra thallus will be emerged. Now moving ahead, this is the details. I have explained everything already. You can pause the video and write if you want. Now the next set of reproduction is asexual reproduction. We have seen that vegetative reproduction was through fragmentation. Asexual reproduction is through a few different kinds of structures. One is chlamydospores or aplanospores. What will happen in this? Actually, asexual reproduction, I told, during unfavorable or extreme kind of situation, the Spirogyra will be going for asexual reproduction. This is the normal filamentous cells arranged end to end. Now during the unfavorable situations, protoplasm, this green color structure is the protoplasm, around which you can see the cell wall. This is the cell wall, and this protoplasm will be separated from the cell wall. You can see there is a gap between the protoplasm and the cell wall. Now these cells are condensed. Everything will be concentrated to the center, and it will be rounding off to a spore. Okay, then this spore will be developing a thin or small wall around itself that will be germinating into new thallus when the conditions are favorable. So this is about chlamydospores or aplanospores. Now coming to the next kind of asexual reproduction, that is through echinids and azygospores. Azygospores are not so important, you can leave that. You can focus on aplanospores that we have already discussed. The protoplasm condensed and rounded off to a spherical structure that is also connected end to end. So this will be breaking off and germinating into new thallus. Now coming to echinids. Can you spot one difference between aplanospores and echinids? Yes, you said it right. In the case of echinids, the protoplasm is not condensed or rounding off. It is laying as such, but the wall will be thick in shape. Okay, in structure, this will be having echinids during kind of unfavorable conditions. During echinids formation, the cell will be secreting a thick wall around itself to resist or overcome the very unfavorable conditions like excess drought like conditions, etc. when water is not available for germination for the new thallus to survive and all. It will be laying as such during unfavorable circumstances, and you can see that this is not the color of vegetative cells. Okay, the echinids will be having a thick dark color with lots of pigments in it, so it could be easily identified and differentiated from that kind of normal vegetative cells. So this is about echinids. So I have already explained the things in echinids. I hope nothing is missed out. So this is about echinids. Now coming to the final and most common mode of reproduction in Spirogyra, that is sexual reproduction. It is commonly done through conjugation. In that, two identical or two different filaments of Spirogyra will be coming together, and it will be forming a conjugation tube. You can see that they came close, and one will be acting like a male thallus and another will be acting as a female thallus, and the two cells which are forming a conjugation tube. Okay, you can see that between the male and female a conjugation tube is formed, and since the contents of one cell is behaving like a male gametes, this particular cell will be called a male gametangium. Okay, now from the male gametangium the entire protoplasm will be transferred to the female gametangium. Okay, you can see then male gametangium became empty. Now this particular female gametangium will be having the nuclei and contents of male and female gametes, and they will be fusing together, and that will be forming a zygospore. This actually that will initially form a zygote, then it will be developing a thick wall around itself, multi-layered wall around itself, and then it will be called a zygospore. You can see the same kind of structure here. This is the zygote which will be finally modifying into zygospore with the development of a thick wall around itself. Now finally the zygospore germinates and the zygospore will be forming a new thallus. Actually both are same figure only, since it is having some illustration I have attached to these things. So if there are two kinds of conjugation, actually one is scalariform conjugation. This is the scalariform conjugation. These both are scalariform conjugation. It is a kind of conjugation when the conjugation tube is formed between two different thalli, where one acts as male and second acts as female. Two different thalli. So such kind of a conjugation is called scalariform conjugation. Now coming to the next set of conjugation, that is lateral conjugation. In lateral conjugation, from the same thalli, different cells, two different cells, this cell acted as a male gametangium and this cell acted as a female gametangium. So the contents or the protoplasm of one gametangium, male gametangium, was transferred to the female gametangium, but both are in the same thalli. Okay, this can happen either like this, a tube formed either like this or either like this, when the septa between two cells are dissolved and a conjugation canal is formed directly within the same thallus. Between the two same cells by just dissolving the middle septa. So this is also a form of lateral conjugation. So this is it. Yeah. Coming to the explanation part, I have already given the scalariform conjugation explanation here and lateral conjugation explanation here. Then finally what will happen in all the case, when the male gametangium transfers its protoplasm to the female gametangium, the fusion will happen and zygote will be formed. You can see that in the zygote, during the time of germination, the zygote will be secreting a wall around itself to form a zygospore, and zygospore will be waiting till the unfavorable situations are over. The sexual reproduction will be happening during the unfavorable circumstances. Okay, normally vegetative reproduction happens during the favorable circumstances, and most commonly during the unfavorable circumstances sexual reproduction will be happening, and this is a more common method of reproduction in the world of Spirogyra. And during the time of germination, this particular zygote or zygospore nuclei will be doing meiosis. Okay, will be undergoing meiosis or reduction division, and 4 haploid cells are formed. Out of this only one will survive. Okay, this is a very important point, and the surviving particular nuclei will be the permanent nuclei for this particular new thallus, and it will be doing the germination. Okay, this now it is the final phase of transformation of zygospore, and now it will be germinating under the favorable circumstances to a new thallus of zygospore. So this is all about the zygospore. I have given the explanation for what happens to the zygospore here. So this is all about Spirogyra. So here completes the green algae and all the life cycles in green algae. We have discussed everything with simple diagrams for each and every life cycles. Around 6 life cycles we saw from Volvox till till Spirogyra. So more than this, there is nothing required from green algae. Now in the upcoming series, we will be discussing about Vaucaria in Xanthophyceae. Then we will be seeing Polysiphonia in Rhodophyta, etc. So till then, bye. Take care.